Now, the metabolism of glycogen turns out to be relatively simple compared to other metabolic pathways that I've talked about in these presentations. The complexity of glycogen actually relates to its regulation. We see on the screen a depiction of the way in which glycogen is broken down. So glycogen is broken down by an enzyme called glycogen phosphorylase. And in many places, this is abbreviated and simply called phosphorylase. As we will see, there are a couple of different forms of phosphorylase for consideration. Now, glycogen phosphorylase is an interesting enzyme in the way that it breaks glycogen down. Many molecules, like sugars and so forth, are broken down by hydrolysis using water to break bonds. Glycogen phosphorylase does not use water. Instead, it uses phosphate, as you can see here. And so this is, instead of being called a hydrolysis, is called a phosphorolysis. Now, that's not just an important thing of nomenclature, but rather it actually is saving the cell energy, as we shall see. The phosphorolysis of glycogen results in production of a molecule called glucose 1-phosphate that you can see on the lower left, and a glycogen that's been reduced by one residue. So what has happened in this catalytic action is that the enzyme has broken the bond, the 1,4 bond, between the end of a glycogen chain. Now, it's important to recognize that glycogen phosphorylase works only on ends. It doesn't cut in the middle, it works on ends, and it sequentially chews its way into a glycogen. So one of the reasons that the branchedness, as it were, of glycogen is important is because that the more ends there are, the more glycogen phosphorylases can start on ends and sequentially release a tremendous amount of glucose very quickly. We hear stories about somebody who's in a terrifying situation. They do incredible feats of lifting something heavy that would seem otherwise impossible. And the ways in which this actually happens because of the branchedness of glycogen, which releases this burst of glucose that people use to accomplish what they do. Glucose 1-phosphate is readily turned into glucose 6-phosphate by the enzyme phosphoglucomutase. Now, this is a reversible reaction, as you can see. The significance of the reaction, though, is that glucose 6-phosphate is produced. And if you remember, we started with a glycogen that had glucoses that had no phosphates on them. We have at this point an intermediate in glycolysis, glucose 6-phosphate, that does have a phosphate on there. And that phosphate got on there not by the use of ATP. If you recall from the glycolysis presentations, hexokinase uses an ATP to put a phosphate onto glucose to make glucose 6-phosphate. Here, this phosphate got put onto the glucose from the glycogen by using a phosphorolysis reaction that did not require ATP. This saves the cell energy. And the energy for putting that phosphate on actually came from the breaking of the 1,4 bond by the glycogen phosphorylase. So cells are very efficient. Cells use whatever they have available to them to do things as efficiently as possible, and this is a very good example. Now, glycogen breakdown proceeds, as I said, by the action of glycogen phosphorylase working from ends. And I also noted that glycogen is a very branched molecule. We can see on the screen here a branch of glycogen and learn a little bit about the way that glycogen phosphorylase works. You see on the top the branch, and you see the end of the molecule where we have 10 different blue residues that are shown on the left. Those 10 residues are all targets for glycogen phosphorylase. So as the enzyme acts, it goes through and it releases those 10 sequentially in the reaction that I showed at the beginning of this lecture. It takes 10 phosphates, and 10 glucose 1-phosphates are produced as a result. Now, at that point, glycogen phosphorylase won't go any further. So glycogen phosphorylase has a limit in how close to a branch it will work. You note that the branch is at the base of the yellow molecules shown on the very top. Now, as we move down to the second part, you see we've changed the color of those glucoses so that 3 of them are still in yellow, and 1 is now shown in the sort of orange color, and there's a reason for that. There's an enzyme called Debranching enzyme that plays the second role in breaking down glycogen. Debranching enzyme takes the 3 yellow residues that you see in the square and transfers them down to the chain below. So because glycogen phosphorylase can only work within about 4 units of a branch, this debranching enzyme plays the important role of rearranging the molecule so the glycogen phosphorylase will have additional substrates to work on. Debranching enzyme is also interesting in another respect, and that is it actually catalyzes two different reactions. The first reaction you can see on the screen, which is the transfer of the 3 residues of glucose down to the lower chain. What's left behind is that single molecule that's shown in the above the main chain. That single molecule has that alpha-1,6 bond that's essential for that's part of the glycogen branching. That last molecule is removed by a hydrolysis reaction. So water is actually used to release that, and glycogen debranching enzyme catalyzes that reaction. So it's a very flexible, it's a very remarkable enzyme in doing what it does, but because of that, you can see that an unbranched form is now left behind, and glycogen phosphorylase can come back in and continue the phosphorolysis that it was doing originally. I should also note one other point here, and that is that the release here of glucose is the only place in glycogen metabolism where free glucose is released. All other releases are glucose 1-phosphate.